Okay, so this is a hard class, yes? But it is not impossible, yes? How are you going to succeed in this class? You are going to study, and you are going to study on a regular basis. A little bit every day, or at least several times a week, yes? In order to succeed in this class, that's what you have to do. Your next test, your first test, is coming up. And I just want to give you the heads up, that's probably one of your easiest ones. Some of you won't feel that way after you take it, but that's what it is. So, if you haven't already started to study for your exam, you start today. I'm pretty loud, so I think you got that. Yes? Did everybody get that? If you didn't get it, go listen to the recording again. Yes?
When we take the test, are they like clickers?
No. Paper. Yep. Clickers are just there for, kind of fun and for attendance, but they're not working for some reason, so we'll see if it works today. We hope it works today. Okay, so let's finish up Chapter 3. Any questions about Chapter 3 so far? So everybody knows everything there is to know about Chapter 3. Right? Because you don't have any questions for me. Good. So let's go on. Plasma membrane. What's it made of? Phospholipids? Give me some other stuff. It's not just phospholipids. Cholesterol, glycolipids, proteins. Yes? Understand all that stuff. We also have cell-to-cell connections that we left off talking about last class. Not only for keeping cells together in a group to form tissues, but also for what? Yeah, so cells can recognize each other. And which cells am I talking about when I say cells recognizing each other? We talked a little bit about this last class. No? Why do I have to recognize, or who has to recognize your cells as being yours? Yeah, white blood cells, your immune system. And then we also have to communicate from one cell to the next. Remember that heartbeat example I was talking about? Is that ringing a bell? To anybody? No bells? Maybe a whistle? Something? Nothing. Cells have to communicate with each other in groups. So the cells that make up the tissue that makes up your heart muscle, for example, has to be able to send chemical messages in a wave down the line to all of its little cell friends to cause a contraction and relaxation of that group of cells. So what kind of cellular junction is going to allow for communication from one cell to the next? Watch this. Did you hear me back there? Did you hear that, what I just said? No. How come? I'm just saying, you answer my questions, some of you, and you go, I can't hear you. Speak up. Nobody knows which cellular junction. Gap junction, very good. You were getting me a little nervous, just a little nervous. All right, and the next thing they talk about in your textbook is something called a cell adhesion molecule, or CAM. And these are protein-like structures that are going to allow for communications between cells. All the things that we just talked about. The other thing they talk about in your book are the roles of plasma membrane receptors. And again, we've talked about this. Some of them are for contact. But some of them, and if we look at this diagram here, which we can find on page 82 in your textbook, talks about a different kind of signaling. Signaling that goes on within the cells, and it's sort of a chain reaction of events that create just change within the cell. The little guys that are running around up top, what are they doing? The little ligand and the receptor and the G protein and the enzyme and the second messenger. You see them at the top of the diagram? What are they doing? Yeah, they're passing information. It looks like they're running what? They're running a little relay race where they're passing the baton to the next player that will then carry on and do what? Pass the baton to the next, and so forth. So when we talk about this type of reaction, it involves several different groups of chemicals, chemical signals, that act like a relay race, or sometimes I like to refer to them as a series of dominoes falling. One has to fall to hit the other, in order for them all to fall in a line, right? So these chemical reactions, these chemical signaling mechanisms, and we're going to talk about them throughout the semester. We'll talk about them when we talk about vision. We'll talk about them when we talk about how hormones work. They are chemical messages that get handed down the line that cause change within cells. So that's what they're describing when they describe all of the players in this chemical messaging group or signaling group. I'm not going to make you memorize them right now. I just want you to be familiar with some of the different terms. There's some sort of receptor on the surface of the cell, and you can see that all the way over on your left. Something comes in contact with it. I'm going to call it a something because it could be a whole bunch of different things. It could be a chemical dissolved in saliva. It could be a chemical dissolved in mucus. It could be a hormone. So the something we're describing here is a ligand. When it comes in contact with the receptor, it's going to start the race. So we see it up in that, again, looking at the little guys running across the top of your page. The ligand would be everything that I just described, one or the other. And the receptor is then going to start the race. It's going to kick off and hand off to something called the G protein. And the G protein is going to get on the move. When it comes in contact with other things, it's going to eventually result in change within the cell. So in this case, we see our G protein on the move, handing off to some sort of enzyme. Now, once that enzyme is activated, what do enzymes do for a living? They make things happen. They make chemical reactions happen at a faster rate. So once that enzyme has been activated, it's going to cause something to happen. In the case of this chemical signaling pathway, it's going to cause something to be formed called a second messenger. That second messenger is then going to keep on going and cause more change. It's actually going to change some proteins within the cell and activate them, causing them to do different work. Again, when we talk about these guys, we're going to talk about them in more detail, mostly next semester. But it's a series of chemical events that will cause change within the cell. And that's how a lot of hormones work. When we talk about hormones, we're going to talk about water-soluble hormones. And we're going to talk about a second messenger system again. So we'll see these terms come up again. All right, the cell. By the way, cells can also be triggered to do things or change by electricity. And when we talk about the nervous system, we'll talk about voltage-gated channels, changes in movement of ions across membranes. I want to throw that in because it does mention it in your book as well. All right, the cytoplasm. What's a cytoplasm? of a semi-squishy, watery environment. And what's inside of it? Little parts. Little parts inside what? Where does the cytoplasm live? Inside cells. So when we talk about cytoplasm, we're talking about the environment we're going to find inside cells. Now, there's a liquid environment inside cells, inside the cytoplasm, we call cytosol. And then there's little parts, little cellular parts. Another name for those little cellular parts are what? Organelles. Do you know what the word organelle means? Little organs, exactly. And each of those little organelles has a certain job to do within the cell. So when I think of cells, I want to think of them as factories. Or think of them as a business or a building, just like Central Maine Community College. Central Maine Community College as a whole would be like the cell. But then each classroom and each instructor and each office has a very particular job to do so they can run this college, right? That's what we should think of when we think of cells. Each of these organelles has a very particular job to do to run the cell on the whole. The other thing we're going to find inside the cytoplasm is something called an inclusion. And that includes things like chemicals, chemical compounds. Some of those guys we just mentioned in that little signaling mechanism would be an inclusion within the cell. Sometimes we're going to find large molecules for energy storage, like glycogen, and those we would call inclusions. So the next part of the book is going to go on and describe all of the different parts of the cell and what they do for a living. So guess what the first organelle is? Mitochondria. So what's a mitochondria doing for me? Yep. Sometimes you'll hear it referred to as the powerhouse of the cell. And why do we refer to it as the powerhouse of the cell? Because this is where a lot of that energy molecule that I'm going to use for my reactions of metabolism are produced. It's not the only place, but the majority of them within the cell are going to be produced here. The little folds we see, or cristae, house a whole bunch of different proteins. And those proteins act like little pumps that are going to help me pump out what? ATP. So when I say the energy molecule, that's the one I'm talking about, ATP. Give me the proper name for that. Adenosine triphosphate. What kind of organic molecule is it? What kind of organic molecule is it? Maybe you didn't hear me. There's 66 people in here and nobody knows what a ATP is? That's going to be on your test. No. Huh? I'm a mother. I can hear everything. What kind of organic molecule is ATP? I got time. Yes, it's a nucleic acid. Thank you so much. See, that's why you're supposed to be studying already. So, most of our ATP is produced in the mitochondria. When we second semester talk about a very complex reaction called cellular respiration, we're going to revisit this guy again. Because the end part of this big, huge reaction called cellular respiration, which we're going to study second semester, you all look so excited. happens here. That is called oxidative phosphorylation, or the electron transport chain. And in those little folds and membranes, in those proteins that I mentioned, that's where it happens. So, it's very complex, a lot of membranes, a lot of enzymes, a lot of proteins, a lot of chemistry going on in the mitochondria. Who's next? By the way, all these pictures are coming from where? All the pictures that are being pulled out and thrown up here. They're inside where? Remember that diagram I told you? See it at the top? It's in black and white. And then the only thing that's color is the organelle they're talking about. Remember I said you have to know that one? That's where it's coming from. So who's next? The ribosomes. Well, in the book that's next. But there's a whole bunch of stuff in there, so yeah, you're right. Remember, these kinds of cells are what kind of cells? There was two kinds of cells I described to you. Very simple cells, and then very complex cells. These are eukaryotic cells, very complex cells. So all of the organelles are what? I'll give you a hint. Nuclear envelope wrapping around the endoplasmic reticulum. They're membrane bound. So the organelles in complex cells, like your eukaryotic cells, all kind of wrapped up in membranes. So what we see here in this diagram is a couple of different ones. But in the book they describe the little dots that are kind of hanging out on the blue portion of these cells. The blue portion of the cells are basically little canals. And there's two kinds of these little canals. There's a whole bunch of little canals that don't have any dots on them. What do they call those guys? Smooth. And the canals are called, you ready for this one? Endoplasmic reticulum. And then we have these same kinds of canals with a whole bunch of those little dots on them. Well, what are the little dots? They're ribosomes, and they're actually little bits of nucleic acid called ribosomal RNA that are going to help us assemble our proteins. So ribosomes are said to be the site of protein synthesis. Now, where do I get the building blocks for my proteins? From the DNA. And where's the DNA? In the nucleus. I'm actually only going to get the information as to what I need to put together from the nucleus. But I'm going to put that together on the ribosomes, specifically on the rough endoplasmic reticulum portion of the ribosomes. Because the proteins that I make will then go into those canals, the endoplasmic reticulum, and get transported different places within the cell. You with me so far? So we have some ribosomes that are just kind of hanging out in the cytosol or cytoplasm, and then we have some ribosomes that are hanging out directly on the endoplasmic reticulum. Those guys are called membrane-bound ribosomes. They're stuck to another membrane. Again, ribosomes important for what? Protein synthesis. Next organelle they describe in your textbook, endoplasmic reticulum, or ER. There's a rough kind, and that's the one that has a whole bunch of what on it? Ribosomes. And there's a smooth kind. No ribosomes. So what happens? These are basically transport for the cell. So they're like the hallways at Central Maine Community College. where I come out of my office and I walk down the hallway, and then, of course, I have to walk across a parking lot and come to this building. So it's a way to get from one place to another, right? But also what's going to happen inside the endoplasmic reticulum, both rough and smooth, is chemical reactions. We're going to bring things together. We're going to pull things apart. So when I need to do something with fats, for example, break it up, what are fats made of? What are they made of? What are the building blocks of lipids? No. Glycerol and fatty acids. Very good. So when I have to rearrange things or put things together or manipulate things, we're going to see things like that happening in the rough and smooth endoplasmic reticulum. Another of the very important players in the cell is something called the Golgi apparatus. And the Golgi apparatus, we're also going to have some chemistry going on there, but one of the big things that happens in the Golgi apparatus is we're going to transport, package, and ship, I like to call it. Sometimes I refer, just me, I don't know if the book does, but for years I've called the Golgi apparatus the UPS system of the cell. What does it do? Package, ship. So it takes certain things within the cell, maybe that have been manipulated in the endoplasmic reticulum, and packages them. Packages them in what? Do we have like brown paper and packing tape inside the cells? What do the cells package things in? Little vesicles, little packages called vacuoles or vesicles, and we'll call them a whole bunch of different things. But what is the package made of? Any guess? Yeah, membrane, phospholipids. So that's what I wrap around these different packages. And for many different reasons as we go along, you'll see these different packages have to be separated from the rest of the cells. For example, what if I make a package full of chemicals that are going to dissolve other things? Do I want those chemicals just floating around in the cell? Because what are the chemicals going to do if they're just floating around in the cell? Dissolve your cell, right? So I have to package them. So the Golgi apparatus is going to help me package and ship. Remember we talked about endocytosis and exocytosis? Who made those packages? Somebody like this. And they're membrane-like packages. So things can come into the cell and things can leave the cell. So when we're talking about things leaving the cell, we're talking about something made by who? The Golgi apparatus, possibly. So in your textbook, bless you, they go on to talk about peroxisomes, which are basically packages of peroxides. Peroxides are going to help break things down. Did it ever get a cut and get some hydrogen peroxide and pour it on the cut? What happens? It makes bubbles, and the bubbles explode, yes? When the bubbles explode, think of that as sort of like a landmine in the little bacteria world that's around it. When they explode, what's going to happen to the bacteria? They're going to get blown up. Yep. So through these oxidation reactions, bubbles that form are going to cause things to break down. And in this diagram, we can see this on page 86, sequence of events from protein synthesis on the rough endoplasmic reticulum to the final distribution of proteins. We're going to talk about a process called transcription and translation. And the packaging of those proteins and shipping of those proteins are going to happen here with the help of the endoplasmic reticulum, handing off some information from handing off information to the Golgi apparatus. So you can see in this diagram, way up there at number 1, proteins are being produced in the endoplasmic reticulum. They're being fine-tuned, and then they're being handed off to the Golgi apparatus. The Golgi apparatus will then package and ship. And this could be proteins, maybe some of those proteins that are on the surface of your cell could be delivered by the Golgi apparatus as well. Lysosomes is another thing they talk about. And all these little packages, it's very hard to distinguish the differences between them. So peroxisomes and lysosomes, they all kind of look the same within the cell, unless I have some sort of fancy chemical stain that can bring them out by coloring them a very specific color. So lysosomes, guess what they do? Or guess what they have in them to help them do? What's the word lyse mean? And it's not those little bugs that get in your head and cause all kinds of problems. When we think cells, what does lyse mean?
Oh, that meant,
Yep, break or burst, right? We talked about that when we talked about tonicity, didn't we? Okay. Exactly. So lysosomes, guess what kind of chemicals they have in them? Things to break things apart or break things down. So, another thing they describe in the book is something called the endomembrane system. And this basically describes all of these membrane-bound organelles within the cell. The plasma membrane is part of that as well. But we also have membranes around all of these little channels, endoplasmic reticulum, the Golgi apparatus, membranes around the mitochondria, and then a very special membrane around the nucleus. It's kind of the Fort Knox of membranes. Within that membrane, which we call the nuclear envelope, we have to have little spaces for things to pass in and out, called nuclear pores. And these are some examples of the endomembrane system within a, what kind of cell is this again? Eukaryotic cell. The next thing they begin to describe is something called the cytoskeleton. And this is a series of different strength proteins that are going to help give the cell its toughness, keep its shape. Starting in your textbook on page 88, and this is figure 3.23, we see some of these different elements of the cytoskeleton. Microfilaments, intermediate filaments, and microtubules. A couple of different things. A, shape. It's going to help the cell keep its shape. Anybody ever go camping and put up a tent? Yeah? It's really hard to sleep in the tent if you don't put it up right. Yes? You know what I'm saying? Those ridiculous poles. You know, this one fits into that one, and you got to get them just right. Well, think of those poles when you think of the cytoskeleton, because they are going to allow the cell to be nice and full so it can do its work, so the tent doesn't collapse on the group. Yes? So microfilaments, and these are awesome, wonderful stains. We won't see anything like this under our microscopes. Unfortunately, we don't have these awesome, wonderful stains and crazy microscopes, but you can see them throughout the cell. When we talk about microfilaments, a lot of times we're talking about movement of things within the cell or movement of the cell itself. When we talk about muscle contraction and something called the sliding filament theory, guess who we're going to mention? These guys. They give us an example of something called actin, or the thin filaments within the cell. And these, well, along with some other proteins called thick filaments, are going to allow the cell to shorten and lengthen. That's what allows you to move. Intermediate filaments, again, acting as a framework to help keep structure within the cell. Some of the things that we see intermediate filaments in are some of those cellular junctions that we talked about. Desmosomes, for example. We'll see some of these filaments creating part of those. And then microtubules. They're the big kahuna of the intermediate, of the cytoskeleton. You can see how thick they are. They're going to help the cell with cell division. So they're big and they're thick. They might also help the cell with some sort of cellular movement. Sometimes cells can move things across the surface. Sometimes cells actually have to swim from one place to another. And we're going to see microtubules as part of that. So we see this little diagram on page 89 in our textbook describing some of the different functions of these cytoskeletal elements. Microtubules and microfilaments function in cell motility, helping the cell to move or helping to move things through the cell as well. If I take a whole bunch of microtubules and arrange them very tightly, I can form little extensions of the cells. When we talk about things like cilia, which are little extensions of the cells, kind of like little hairs at the top of the cell's head, they can actually wave and move and push things across a group of cells. Somebody just went and they swallowed. You know what happened? Not to gross you out or anything, but lining your trachea, you have mucus. And the cells that line your trachea have those little cilia on them. And what are they constantly doing for you? Yeah, when you breathe in, unfortunately, we don't all breathe sterile filtered air, do we? So there's little, and this is my famous word, schmutz, little pieces of stuff, dust, in the air. Do we want those way down in our lungs? No. So the mucus is going to help by trapping some of the stuff that I'm breathing in through the air. And once it's trapped there, where's it go? The cilia on those cells are going to wave that mucus up, away from the lungs and towards your what? Towards your mouth. And what are you going to do with that mucus? Swallow it. You swallow it all day long. Except if you're a baseball player, then you spit profusely. But most of us swallow it all day long. It wouldn't be socially acceptable for us to spit in the classroom. People will get a little upset about that, right? Why do you swallow it? Yeah. The mucus will go into your stomach along with all the little schmutz that got trapped in it. And what's going to happen to it? It's going to hit an acid bath. Hydrochloric acid, to be exact. And what's going to happen to any little nasty particle that might be in there? Digested. Yes? That's cilia. So these guys have to allow for movement. A little bit tougher, a little bit more organized. And they will cause, through a series of movements, like these little protein movements they described before, movement of the cilia, which will cause waves to form, which will cause things to move across the surface. Any smokers in the group? Nobody smokes in this room. Liars. There's got to be a smoker in here somewhere. No, is there? All right, good. Gee, phew.
I was trying to be polite about it.
I know, that's all right. One person admits that there's probably a heck of a lot more than one in this room. What happens when you inhale smoke? Do you know? besides bringing down the schmutz that you're trying to keep out into your lungs, you also paralyze those guys. Only for a period of time, about 15, 20 minutes every time you smoke a cigarette. And it's cumulative, by the way. So you're still breathing in and out, right? And now you're smoking, so you're breathing in and out lots more schmutz. That's all getting stuck where? In the mucus. And as it sits there for long periods of time while the cilia try to recover, it's going to fester. If it's bacteria, it's going to have a heck of a time reproducing itself. Yes? Sitting there on the nice lining your cells, producing all of this toxic substance. And then eventually, when you go to bed, hopefully you don't smoke while you sleep, what's going to happen to your cilia? They're going to wake up, and they're going to furiously push all that excess mucus and all the lovely festering material that's been sitting all over your trachea all day. Where? And you wake up in the morning and you start your day, go have a cigarette, cup of coffee. Starts all over again. How much do cigarettes cost? $8, $9? Think of the money you'll save if you stop smoking. Not only the money you'll save on cigarettes, and that you can save and thank me for having you stop smoking, maybe take me to the Bahamas or something. I'd appreciate that. That would be nice. What else? What else are you saving? Your life, healthcare costs. When we hit our discussion about chronic obstructive pulmonary diseases in the respiratory system, here's a scary little factoid. 80% of all cases of COPD are directly related to what? Cigarette smoke. 80%. Now you guys are the king and queens of Google, yes? Go Google those numbers up a little bit. See how many people that means. Maybe if you see numbers and you say, holy crap, maybe that'll stop. Or make you step back and think about stopping. By the way, I'm an ex-smoker, so I don't want to hear that, oh, it's hard to stop. Yeah, it is. It's really hard. It's very hard. But you can do it. It's not impossible. And think of everything, secondhand smoke. Yes? How many people were really mad when they said, no more smoking in public? How about all those people that had to get exposed to your smoke? Yes? They didn't choose to do that. So, that's my little spiel about smoking. Always get there when the microvilli come in. So, extremely important, microtubules and the arrangement of those microtubules that help movement. There's other things microtubules help with, something called flagella. When we talk about humans, there's not too many flagellated cells. Usually when we talk about flagella, we're talking about a group of organisms called protista. And those are single-celled organisms that float around in the lake. But what cells are flagellated in humans? They have to go from one place to another. Sperm. The next thing they talk about is the nuclear envelope. So a very hardy membrane that surrounds your genetic material. DNA and RNA. What kind of organic molecules are DNA and RNA? Nucleic acids. Ooh. And then something called the nucleolus, which that's kind of the RNA factory that hangs out in the center of the nucleus as well. So the nuclear envelope is going to help protect the genetic material and allow only certain things through the nuclear pores and nuclear pore complexes to go in and out of the nucleus. The nucleoli, again, we're going to see some RNA. These molecules are going to work hand in hand to help make proteins. When we talk about DNA, it's a huge ladder, double helix, of nucleic acids. Now, what are the building blocks of nucleic acids? No. Everybody wants to say protein. Proteins are proteins. What are the building blocks of proteins? Amino acids. Nucleic acid's not a protein. It's a nucleic acid. And what's it made of? Nah. She's got it. It's close. It's there. Phosphates, sugars, nitrogenous bases. If I'm talking about DNA, how many different nitrogenous bases are there? 4. Adenine, guanine, thymine, cytosine. They come together in little units called what? Nucleotides. One nucleotide is phosphate, sugar, nitrogenous base. Yes? So, 4 different nucleotides make up chromatin. That double helix we looked at when we looked at chapter 2. Lots of it, yes? So chromatin is a bunch of different code, basically. Series of these. If we look way up at the top of the diagram, we see the DNA double helix. And then we get so many of these, we have to sort of wind them up and keep them together. And these little proteins called histone proteins are going to help wind up all of this genetic material and keep it linked together. It's kind of like, anybody ever help their grandmother unwind a ball of yarn or make a ball of yarn? You go to the store and you get that. Nobody knits anymore. You get that. What's it called? The skein. Thank you. And you start it, and your grandmother holds on to it, and you make a ball out of it. That's kind of what DNA and histones do for each other, so that they won't get tangled up, so that we can use the DNA material. We can read the code. On page 94 and 95, your book has done a very nice job of giving you table 3.3. And this is all the parts of the cell. Use the tables to your advantage. They're going to help you do what? They're going to help you study. So when we talk about a cell, it's sort of got a life. Just kind of like you. When you're born, you're little. Yes? What do you have to do? You have to grow so that you can do what? Go get a job, make a living, and then hopefully you'll reproduce. So that's kind of like the day in the life of the cell. It's born, it has to grow, it has to do what it does for a living, and then hopefully it'll reproduce. So that's what they talk about when they talk about cell growth and reproduction. And the different parts of a cell's life are described here in the cell cycle. So figure 3.31 on page 97 in your textbook describes or shows us the cell cycle, the life of a cell. G1 is the first part of a cell's life. First of all, that where I'm growing part of my life and doing what I need to do for a living part of my life as a cell, that's called interphase. So the different sub-phases of interphase are G1, that's the growth phase. When I decide it's time to have kiddos, I have to prep as a cell. When I decide to reproduce as a cell, I have to make copies of the map I need to make a new cell. Because every single cell in your body has a copy of the map. When I say copy of the map, what do I mean? That DNA we just looked at. So during the S phase of interphase, what do? I get started copying the map. How many chromosomes do you have? 46. 23 pairs of chromosomes. Millions of bits of information. All have to be copied. So during the S phase of the cell cycle, interphase, we're still in interphase, I'm going to take my 46 chromosomes and create how many chromosomes? Very good math. 92. Yes? 46 plus 46 is 92. And this is where these guys are going to come into play for me. What are they going to do? They're going to help condense all those copies down. I sometimes refer to this as, you know, when you have to move from one place to another. You want to move all your stuff, right? What do you do before you move all your stuff? You pack it up, right? You put it in a box, and you put a label on it, and you pack it up. Why are you doing that? Yeah, condense it down or put it in a manageable place so that when I go to move it, it's not going to all fall out. Right? So that's what these guys are going to help me do. They're going to take the chromatin during the S phase. Now I have twice as much. It's kind of getting crowded inside that nucleus. And I'm going to condense it down. I'm going to turn chromatid into chromosomes. When I look under the microscope, I can actually see them because they're so fat. That's what you're going to do this week. You're going to go find all the different phases of the cell cycle under the microscope. Okay? So S phase is where I replicate the genetic material and prepare for the split. G2, I have to kind of proofread things, make sure everything got packed, make sure I didn't leave anything behind. Kind of like when you go to a hotel, you go through all the drawers, you check under the bed, make sure you didn't leave your shoes or your toothbrush, right? That's the G2 phase of interphase. Getting ready for the mitotic stage of my life. So I come out of the green interphase in the diagram and I go into the mitotic stage of my life. And what's mitosis? Any clue? Cell division. There's different stages, different things happening during the mitotic stage of my life. Prophase. What am I doing during prophase? This is where I'm going to take all my stuff that I have to move in, package it up, condense it. I made copies of everything, right? Why did I make two of everything? I'm splitting into two separate cells. So half and half have to go to my brand new cells of genetic material. During metaphase, I'm going to take the copies and I'm going to line them up. During anaphase, what am I going to do? Split the two copies so they go to opposite ends of the cell. By the way, those cytoskeletal elements, the microtubules, those are the guys that are going to help me do all of this. Once anaphase has taken place, now I can have two brand new cells, right? Not quite, because DNA isn't the only thing my cells have, right? What else do I have to make sure both the cells have? All those different organelles we just finished talking about. So during the end of anaphase and through the last phase of cell division called telophase, a process called cytokinesis, and we see it's kind of a weird looking thing. It kind of loops into the anaphase a little bit. See how it does that? That's showing us that that's when it starts. Cytokinesis is the replication of what? All those organelles. So I'm going to make some more mitochondria, and I'm going to make some more endoplasmic reticulum. I'm going to make some more ribosomes, and I'm going to make some more lysosomes, so that who can have them? Both are brand new cells. So it starts at the end of anaphase and continues through telophase. The book talks about DNA replication. Now, there's a whole bunch of different enzymes that are going to help me with DNA replication. This is a nice little diagram because it gives you a visual. It's sort of like a little knitting machine that's going to help with the production of new DNA. I'm going to use the code to make more code. Remember when I talked about DNA earlier in Chapter 2, I said it was made of nucleotides, and there were 4 different nucleotides: adenine, guanine, thymine, cytosine, different shapes, purines and pyrimidines, right? And purines can't attach to each other. Otherwise, the ladder will be weird shaped. So there's only a very certain pattern that can happen between these molecules that will create a ladder that is the right dimension. So adenine can only attach itself to what? Thymine, to create a rung. Do you see the colors over there on the other side? And guanine can only attach itself to cytosine. Now remember, these little units are called nucleotides. When you eat food, do you eat DNA? Yeah. Name anything you eat that wasn't at one time living. There's only one thing that comes to mind for me. Anything for you? Salt. Everything else, salad, bread, pasta, cheeseburgers, French fries, they were all what? At one time alive. So when you eat it, what do? You break its cells down and use the building blocks for your work. The building blocks I'm talking about are these things. What are these things? Those are nucleotides. I got them from, let's say, French fries. And they're kind of hanging out in my nucleus. When it's time to make new DNA with the help of these amazing enzymes called polymerase enzymes, and when I'm making new DNA, what do we call the polymerase enzymes? DNA polymerase enzymes. I'm going to bring those free nucleotides in. First I have to do what, though, to my original material? I have to pull it apart so I can make 2 copies. And as you can see, the 2 copies end up being exactly the same because I can only put an adenine with a thymine, and I can only put a guanine with a cytosine. Yes? So this is DNA replication. We have a leading strand and a lagging strand. I'm not going to get too crazy about you. That's a genetics class. We actually teach that here, so you can take that if you want. But there's a 3' and a 5' end, and it can only go in a certain order and in a certain direction, so everything doesn't get all tangled up during the replication process. They describe a little bit of that in your textbook under, where is it? It's on page 97, DNA replication. So there's primers that are going to help the unzip process, and then there's polymerase enzymes that are going to help with making 2 new strands. You with me? Then they go on to describe mitosis. Mitosis, cell division, cytokinesis, division of the organelles, or replication of the cytoplasm is what it's referred to as sometimes. There's different molecules called cyclins and CDKs that are going to help regulate the cell cycle. So some cells will stay in interphase for much longer than other cells. For example, these cells here, we're going to talk about them in chapter 4. What kind of cells are these? They're the exterior skin cells. We're going to refer to them as epithelial cells. They're constantly reproducing. Every time I go like this, what happens? Yeah, dead skin cells are falling all over the place. Your mattress, when you buy it, if you had a scale to weigh your mattress, if you waited 10 years later, guess what? It would be a lot heavier. How come? Because all your dead skin cells hanging out in your mattress. I know, I would have grossed you out several times in this class. Not only your dead skin cells, but little organisms that are eating your dead skin cells. Dust mites. Yes? So now you'll go home and you'll get in bed and how do you feel itchy every once in a while? Ooh, it's those dust mites. So, the other thing this process can do is help make proteins, and that's described in this chapter as well. So you should know the mitotic stages, understand interphase. That's not really a mitotic stage, but sometimes we'll throw it in there when we talk about mitosis. It's kind of important because during the S phase of interphase, that's what am I going to do? Replicate my genetic material, and mitosis couldn't happen without that, right? So then we have prophase, early and late, and these again are awesome pictures. We see the nucleus, and what are those little fuzzy things over there? They're yellow. It's a group of microtubules. That's another organelle. It's called the centriole. And they are going to help form microtubules to help with this whole splitting process. There's 2 of them, so what's going to happen during prophase is they're going to migrate to opposite ends of the cell. And then it's kind of like sticky fishing poles. That's the best way to describe it. By the end of prophase, we've produced some of these sticky fishing poles, and we've packed up all of our what? What's that blue stuff that looks like threads there? That's DNA. So by the end of prophase, I've done all my packing. I'm ready with my sticky fishing poles, and I'm going to line everybody up in the next phase called metaphase. Now the copies of genetic materials tend to stick together. There's a protein that holds them together. It's called the centromere. And the sticky fishing poles that are produced by the centrioles are going to help grab onto either end and do what? Pull them apart. So it's really nice. You can see during metaphase there, and again, you're not going to see this under the microscope because we don't have all these beautiful colors. Everything's going to be sort of in the same tone. But you'll see the spindles that are produced, those sticky fishing poles, attaching to the chromosomes. And what happens is they kind of line up like this and sort of look like little spider legs. And then during anaphase, I'm going to reel in my catch. So what happens to those microtubules? They shorten up, and they pull on what they're attached to. So what's going to happen during anaphase? Watch my fingers. See the little legs? See what happens during anaphase? So you can see them kind of hanging, facing each other, but away from each other. They are pulled away to opposite poles of the cells. What else is going to happen at the end of anaphase? I'm going to start replicating the cytoplasm during, what's that phase called? Cytokinesis. And then we have telophase, where we're sort of creating or organizing back. So you moved into your apartment. What are you going to do next? I need the blender. Where is it? You're going to have to go unpack because you have to actually use that stuff now. So you're going to unpack and you're going to reform some of the things that you took apart to move. So say you have a crib at your house and you have to move it to your new house. You have to take it apart, put it in the moving truck, move it, and then what do you do when you get to the new house? You got to put it back together. You disassemble the nuclear membrane during this process. What do you have to do once you get to the new house? Put it back together. So that's what happens during telophase. You also have to form a nuclear membrane between the brand new cells. So you separate them. We don't just want to pull them apart. What would happen to all the innards of the cell? Yeah, they'd all fall out. So we have to create a separation, and that's what this cleavage furrow is, basically creating new plasma membrane for the two new cells that will be created by the end of telophase. Okay, this is the fun part. Protein synthesis. The other very important molecule we're going to find in the nucleus is RNA. Now, RNA is what kind of organic molecule? Nucleic acid. Very good. And what are nucleic acids made of? Correct. Phosphates, sugars, nitrogenous base. What was that called, the three together? Nucleotides. The four nucleotides that make up RNA are not the same. A little bit different. We have adenine, just like we did in DNA, and we have guanine, just like we did in DNA, and we have cytosine, just like we did in DNA, but we don't have any thymine. This other molecule, exact same shape, is called uracil. So those are the four nitrogenous bases that make up RNA. That's what makes it different. Also, what else makes it different? It's called ribonucleic acid instead of deoxyribonucleic acid. The sugars are different. It also doesn't typically or isn't typically found in human cells in a double strand. It's typically found in a single strand. And it is going to help transfer information from DNA to the ribosomes to help with protein synthesis. All right, are we ready? This is a fast ride. Buckle in. We're going to talk about the process of making a protein using directions from the DNA. And sometimes I describe this whole process as a secretary and the boss. The boss sits in his office. Beautiful office, doesn't like to come out. But the boss has a lot of information he has to get out to the company. Remember, this is like a big factory, isn't it? So the boss needs to get information out to the company, but he doesn't like to leave his office. So the secretary does what? She comes into the office and takes a message, writes down what the boss has to say. What's that called? It's called transcription. So the secretary, in my analogy, in the case of making new proteins, the boss would be who? DNA. And the information the boss has to give lies in the order of all of those little nitrogenous bases in the DNA molecule. The order of the A's and the T's and the C's and the G's is the information. So the secretary, in the case of our cell, is going to be RNA. And there's a couple of different RNAs that are going to help with this process. In the transcription process, we have an RNA called messenger RNA. Now I'm going to form that message by reading a portion of the code. Remember, the code is huge. I only need a portion to make a protein. So I have to unzip the genetic material, and that little guy up there is going to help me. He's called RNA polymerase. In the region in which I start, the beginning of the message is called the promoter. The code itself, the information I need to transport, is in the portion of the genetic material called the coding strand. And with the help of this enzyme, I am going to form a strand of messenger RNA. And the strand of messenger RNA is basically, and these are all the steps. So I just want you to look it over, understand that initiation, elongation, and termination are all part of forming messenger RNA. Remember, I'm forming it based on what? The information I get from the DNA. I want to go to this one because I want you to see this picture. So basically what that enzyme is doing is unwinding the genetic material so I can read the code. You understand what the code is, right? The series or sequence of DNA and the order of those nitrogenous bases. And I'm going to make a copy. Now, A's can only fit with U's, because I'm talking about RNA now. And C's can only fit with G's. So at the end, I'm going to have a strand of messenger RNA that's the exact copy, the exact opposite of exactly what I have on the DNA. Who's going to help me with that? Who's that enzyme? RNA polymerase enzyme. And that is transcription. Okay. Whoa. What the heck is that? It's the code, the genetic code. On page 104 in your textbook, figure 3.36, it's the code. It's the map. Now, I'm going to give you guys a big break, because I know right now if I go through this, I'm going to go through this fast, and your brains will for sure explode. So here's the deal. You are going to go home, A, start to study, and B, finish reading the rest of this, because we're going to come into class on Thursday. We're going to power through these last few pages of Chapter 3, and we're going to start Chapter 4. So that means, here's your gift, your present, thank you very much, Maria, you're awesome. That gives you a whole week to do what? Study, because your exam's going to be Tuesday of next week. Heed my warning, my friends. Start studying today. Make sure you sign the sign-in sheet, wherever it is. Yes, hon? I'll open the quiz for Chapter 3 today. You'll have to finish it by Thursday. I want to make sure you study. I'm not going to give you a break. So, I will pick up with transcription, translation, protein synthesis, and you guys will be able to answer all my questions on Thursday.